Chapter 7, Epidemiology. Pathology is the study of disease. Etiology is the study of the cause of a disease. Which organism or organisms caused it? Pathogenesis, the development of disease. How did it start and how did it progress? So when you think of pathogenesis, besides the development of disease, it's the ability to cause disease. Final effects, what damage was caused and why. Infection versus disease. An infection is colonization of the body by pathogens, so an invasion by microbes. It can occur without causing disease. For example, someone can be infected with HIV and not show the AIDS disease for quite some time. We also have pathogens or opportunistic pathogens, and opportunists will be discussed later. Disease is an abnormal state in which the body is not functioning normal. So we see changes in the health and damage to the host. Normal flora or microbiota. Normally, we're technically, with quotation marks around sterile, we're technically born sterile. Normal flora develops the first few weeks and gains permanent residence. So normal microbiota permanently colonizes the host. You have 1 times 10 to the 14 versus 1 times 10 to the 13 body cells. So you have a lot more normal flora or bacterial cells than you actually have of your own body cells. Some of our normal flora are pathogenic. So normal flora versus transient flora. So the pictures below show you examples of where you would find your normal flora, whether it would be in your nasal cavity, in your stomach, or in the small intestine. Transient microbiota may be present for days, weeks, or months, and then disappear. Relationship between humans and microbes. Symbiosis is the relationship between the normal microbiota and the host. There are 3 types of relationships. Commensalism. In commensalism, one organism benefits and the other is unaffected. So example, Corynebacteria in our eyes. Mutualism. In mutualism, both organisms benefit. E. coli in the gut synthesizes vitamins K and B, and our bodies provide the nutrients to E. coli for survival. In parasitism, one organism benefits at the expense of the other. So one benefits and the other is harmed. And this is any pathogen that enters the human body. Opportunistic pathogens. Some normal microbiota are opportunistic pathogens. Here are 3 circumstances for gaining control or disease. Normal flora ending up in the wrong environment. For example, E. coli in the urinary tract or wounds. E. coli is normally found in our large intestine and the colon. However, if we had fecal or bowel movement and we wipe the wrong way, it could enter the urethra. Now you've given it the opportunity to cause a urinary bladder infection. Compromised immune system. So pneumocytitis carinii, which causes pneumonia, and AIDS. We see that because with the AIDS disease, the immune system is immunodeficient, so that means that it's not working correctly. So this bacteria causes pneumonia. Disruption of homeostasis. A secondary infection after the flu. So you have the flu, and then right after that, you might have bronchitis, or you might get pneumonia. So there's a disruption of homeostasis. Microbial antagonism is competition between microbes. It prevents the growth of many pathogens, primarily transient pathogens. How? There's competition for nutrients. Okay? So the normal microbiota protects the host by competing for nutrients. It occupies niche that pathogens might occupy. They produce bacteriocins. Bacteriocins are proteins that inhibit the growth of other bacteria of the same or closely related species. pH, that produces acids. Oxygen, they use up all the oxygen. So all of these are ways that the normal microbiota will protect the host. Here are some examples. Vaginal flora and yeast. Vaginal flora keeps the pH acidic to prevent the growth of yeast. The use of douche and antibiotics lowered the vaginal flora, leading to an increase in pH, allowing the yeast to grow. And then you end up with a yeast infection. In the large intestine, when antibiotics is used, it lowers the GI flora. This leads to the growth of Clostridium difficile, which is responsible for most GI tract infections after antibiotic therapy. Probiotics. Probiotics are live microbes applied to or ingested into the body. Probiotics are intended to exert a beneficial effect. So it's a desired effect. It improves intestinal development. It helps to prevent and treat disease. We need our normal flora. Sterile environments will create sickly organisms. And this is a picture showing you some examples of some probiotics that are on our store shelves. Recall at the beginning of chapter 7, we said that etiology was the cause of disease. The germ theory of disease states that most infectious diseases are caused by germs. In 1876, Robert Koch proved that microorganisms caused disease. He discovered Bacillus anthracis, or anthrax, in cattle. He established a sequence of procedures called Koch's postulates to prove that a particular microorganism causes a particular disease. So Koch's postulates are used to prove the cause of an infectious disease, and is still used today. Some pathogens can cause several disease conditions, and some pathogens cause disease only in humans. So let's take a look at this sequence of procedures called Koch's postulates. If we take a look here, we'll see in the figure that we have a dead mouse. He took microorganisms from the infected animal and then injected it into a healthy animal. But what did he do before he did that? Well, he took it and he made a pure culture, and then he also identified it under the microscope. So you see here we have some bacillus, and we could say they're in chains, so strep bacillus. And then you see that we have a pure culture. He then took that pure culture and injected it into the healthy animal. Now, what should happen to that healthy animal? That healthy animal should die, right? If it's the right disease that we're trying to identify, it should cause death in the host. If it doesn't cause death, then we're not looking at the correct disease. So we take it from that healthy animal, and then we do the same thing we did in the beginning. We're going to make a pure culture, and then we're going to make sure it looks exactly the same under the microscope. So you see again, we have the same exact thing. We have strep bacillus. Then we're going to do it one more time. We're going to take it and we're going to inject it into another healthy mouse. And hence, if we have the same result, then we know we've identified the cause of the disease. So in summary of Koch's postulates, the same pathogen must be present in every case of the disease. The pathogen must be isolated from the disease host and grown in pure culture. The pathogen from the pure culture must cause the disease when it's inoculated into a healthy, susceptible laboratory animal. The pathogen must be isolated from the inoculated animal and must be shown to be the original organism. There are some exceptions to Koch's postulates. For instance, if you had something that was an obligate intracellular parasite, such as Treponema pallidum, which we know causes syphilis, it cannot grow on artificial media, so for us to create a pure culture, we'll not be able to do so. The same disease, symptoms, but then it could be caused by different organisms. For instance, pneumonia. I know you guys have heard throughout the semester I have mentioned several organisms that cause pneumonia. So you can have the same pneumonia symptoms but different organisms. You can have the same organism but then it causes different diseases. For example, Streptococcus pyogenes. And then finally, the ethical issue of postulate number 3. What do you think the ethical issue of postulate number 3 is? Correct. We are using healthy animals. So PETA does not like this. Classifying infectious diseases. A symptom. A symptom is a change in the body function that is felt by a patient as a result of disease. So a pain. So that's felt by the patient. And they tell you on a scale of 1 to 10 how they feel. A sign is a change in the body that can be measured or observed as a result of a disease. So we can measure someone's fever, right? So we could tell if someone has a fever. A syndrome is a specific group of signs and symptoms that accompany a disease. Communicable disease is a disease that is spread from one host to another. So, for example, chickenpox. Chickenpox spreads from host to host. A contagious disease is a disease that is easily spread from one host to another. So you'll see here, there's a big difference between communicable and contagious. Contagious is easily spread, okay? Whereas a communicable disease like chickenpox, as long as you're not touching it, you can't get it. Non-communicable disease is a disease that is not transmitted from one host to another. For example, tetanus. Tetanus is a non-communicable disease in which the bacterium is introduced into the body. Prevalence. Fraction of the population having a specific disease at a given time. So it's the range. So the prevalence of HIV in 2008 was 1,178,350. Incidence is the fraction of a population that contracts the disease during a specific time. So it's the spread. So the incidence of HIV in 2006 was 56,300. The incidence of HIV in 2007 was 48,100. So in 2006, 56,300 people contracted the HIV virus. Okay? I have a link here that you can visit about HIV, and you can go there and you can read about it and see if it will, work for you. But that's the link there. Occurrence of diseases. A sporadic disease is a disease that occurs occasionally in a population. So, for example, plague. An endemic is a disease constantly present in a population. So, a cold. Colds are present whether it's fall, spring, winter, or summer. You can catch a cold. An epidemic disease is a disease acquired by many hosts in a given area in a short time. So many people in a given area acquire it in a short period. So flu season. Okay, So during the flu, a lot of us get it. A pandemic is a worldwide epidemic. So it's an epidemic that occurs worldwide. So for example, swine flu. Severity or duration of a disease. An acute disease, the symptoms develop rapidly. So for example, the flu. With the flu, you woke up this morning, everything was feeling alright. By the end of the day, you don't feel good at all. Chronic disease, the disease develops slowly and the reactions are less severe over long periods of time. So someone that has hepatitis don't know they have hepatitis because it's developing so slowly. A latent disease is a disease with a period of no symptoms when the causative agent is inactive and then becomes active later. So for example, shingles. So if you had chickenpox before when you were younger, you now have this latent virus laying inactive at the time and then it becomes active later. Herpes is the same thing in HIV. Immunity. Immunity lowers the rate of spread. It lowers the incidence. We do vaccinations to increase immunity. So we see this with smallpox, and now that we have a 0 incidence, we don't have anyone that has contracted it at all. Herd immunity is immunity in most of a population, and technically it's usually 70% immunity in a population. Host involvement. A local infection, pathogens are limited to a small area of the body. So the invaders are in a small area. So for example, boils and Staphylococcus aureus. So if you ever had a boil, you know that it stays in one spot, and that's a staph infection. Systemic infection is an infection throughout the body. So it's either spread by lymph or blood, and measles is a perfect example of this. A focal infection is a systemic infection that began as a local infection. So it's local, then it enters the lymph and travels to other specific areas. So for example, bacterial endocarditis. This is for my dental hygienist to be. It's very important when you're cleaning someone's teeth to find out if they have any heart conditions that exist at the time, because when you're cleaning teeth, you're removing all that bacteria, and if they swallow it, it could travel to their heart and cause bacterial endocarditis. Sepsis. Sepsis is a toxic inflammatory condition arising from the spread of microbes, especially bacteria or their toxins, from a focus of infection. So bacteremia is bacteria in the blood. Septicemia is the growth of bacteria in the blood. Notice the difference. Bacteremia is bacteria in the blood, whereas septicemia is the growth. So there's bacteria actually growing in the blood. Toxemia is toxins in the blood. Viremia are viruses in the blood. A primary versus secondary infection, and acute versus opportunistic. A primary infection is an acute infection that causes the initial illness. A secondary infection is an opportunistic infection after a primary infection. Subclinical infection is when you have no noticeable signs or symptoms. So it's an inapparent infection. So again, hepatitis A. You don't know that you have hepatitis A until later on. Here ends Part A of Chapter 7.